I'm Abe.
I'm Bao.
And I'm Callaway.
And we're gonna and our topic was on the first day. So, a nanometer is a unit of length in the metric system. It's equivalent to a billionth of a meter, or 10 to the negative 9 of the 10 to the negative ninth power meters. Normally the nanoscale ranges from 1 to 100 nanometers in size. Every day we use nanostructures ranging from sports to medicine and engineering. To give you an idea of its size, viruses can be as small as 20 nanometers. Light diffraction is a way that these types of nanostructures could be studied. Light diffraction is a phenomenon that occurs when light encounters some sort of obstacle or slit. In the experiment that we did, we shined a laser through a diffraction gradient of at different heights and measured the distance between the points shown. Through the diffraction, if the point could be seen, it was something called constructive interference, and if the point did not appear, it was called destructive interference. And now Pao will explain the picture.
Can I get the laser? Yeah. Okay. So in that part over there, we can see a lot of people going out of this dance party, I think, and like when they are getting out, they are dispersing. So this is what happens when light encounters a slit. So whenever it encounters a slit, depending on its wavelength, it will bend in all directions. So it will like scatter around and go in all different directions. Also, on the disco ball, we can see the reflection of the lights. So whenever the light hits the disco ball, it's not a slit, it's an object, so it reflects what the disco ball, like the nanoparticles mostly, they reflect the light, or they show different patterns that they have inside. And also at the very bottom, we can see two guys dancing. This reflects the destructive interference and the constructive interference. In the part they are dancing together, it's a constructive interference because they are dancing at the same rhythm. And in the second part, they are doing the destructive interference because they are not dancing in the same rhythm, and the girl tripped, or the guy made her fall.
And then, despite that, there is some parts of this analogy that don't tie the scientific concepts together as well. I think one of them is the disco ball, which is, as you see here, it's kind of creating a diffraction gradient, which disco balls don't normally do to display their light. So I think this one has some type of structure, if a nanoparticle on its surface, which is creating it to make these diffraction patterns. And also, I think that this analogy could have better incorporated the constructive versus destructive criticism or interference shown at the bottom, and has somewhat display that throughout the light. Whereas, as you see at points like here, here, and here on the disco ball, they're examples of constructive interference, since the crests and troughs of the wavelengths are meeting up. So that's amplifying the effects and makes you able to see the light patterns, whereas spots in between the two, between them where you can't see the pattern, it's having destructive interference. So that means that the crest of one light that's being diffracted is meeting the trough of another light. And that results in the wavelengths canceling each other out, and then you can't see the light in between them. So I think that brings us to questions.
All right, so I've got a question. So you mentioned that that when it encounters an object or a slit, depending on the wavelength, it might diffract. So can you elaborate a little bit more on that? So what do you mean by depending?
Okay, so okay, I got it written down. So diffractions is the slight bending of light as it goes through a slit on an edge of an object. And also this bending is determined by the wavelength of this light. So what I'm saying is, depending on the like closure, I think, of the slit, it can also like bend in different certain ways, but mostly because of the wavelength, it will bend more or less. Like it will scatter more, like wider, I think. So
So
Yeah,
right, so
I'm-
diffraction is occurring when light encounters an object or a slit that is.
on the same wavelength as the object is.
Yeah, an object or a
similar.
slit on the same length scale as the wavelength. And what is the wavelength of light?
From 300 to 700 nanometers.
Yeah, approximately.
So any other questions?
I will sit here awkwardly until a parent asks a question. Although Andrew has one too, so we will let Andrew ask one. But I will make every round of students have a parent question. But Andrew, yeah, go ahead.
So if you look at the nanoscale, like tens of nanometers, what type of light would you need to do diffraction with?
Can you repeat that? I didn't hear it.
So if you're looking at nanoscale objects like tens of nanometers, what wavelength or what type of light would you need in order to identify diffraction patterns?
Okay, so that day we did, I think we went down and did and they explained us a way to reduce the scale of the wavelength, reduce it to half of it, but I don't know about 10 nanometers, so probably we would use like, I don't know how to explain it, but it's like a sensor in a microscope that goes like line by line. I don't know how to explain it.
Yeah, I think what he's trying to say is you could use waves that are at the length of the holes of the 10 nanometer, and then use some type of sensor to pick the diffraction. So whereas with diffraction with white light that we can all see, we can use our eyes or a microscope to do it. But with something like that, we would use smaller wavelength on the electromagnetic scale, and then we could use sensors to pick up the diffraction.
Yeah, I think some of the talk you listened to was about X-ray diffraction, so you would use X-rays a lot of the time.
Or you can also
Use the scanning electron microscope.
Yeah, great. So you can also use electron diffraction. So diffraction doesn't just occur with light, it occurs with any type of electromagnetic radiation. Or any type of wave, really. You can diffract with water, with all sorts of stuff. Parents. Or students. I will make one of you ask a question, and I will wait. Excellent.
Yay.
Well, you mentioned about nanomolecules like viruses or bacteria. Say virus doesn't have a cell membrane. Okay, how do you detect virus using any kind of wavelength or using imagination? How do you make any kind of, using light, the method you just described to detect virus and find its properties?
Well, I think you could use like wavelengths as the X-ray diffraction, like at the length of the virus, and you could use that to kind of figure out the properties.
When you guys were at the, remind me, when you guys were at the electron microscopy demos, either at the scanning electron or the transmission electron microscope, did they talk to you about cryo-EM, cryo-electron microscopy? Or did they talk about some of the more complex structures like proteins that you can use, that you can analyze with X-rays?
I don't believe so.
Yeah.
Okay, so so just to answer a little bit more your question, since it is an interesting one. So you can do what's called protein crystallography, where you use X-ray diffraction to try to resolve really complex structures like proteins by trying to make sort of crystallized versions of those proteins. But one thing that scientists are doing now is instead of using X-ray diffraction, because that's a lot more complicated, is something called cryo-electron microscopy, where you basically like freeze the virus in glassy ice, and then you put it under an electron microscope, and you view it with a bunch of different angles, and then using software, you can sort of resolve all of those different pictures into one three-dimensional image. And so that is how you can use these types of techniques to get images of really, really small things like viruses. Any last questions? And if not, let's thank our speakers. Nicely done.